Alright, welcome once again to my class. Today we're talking about mitosis, meiosis, and the genetic code. In mitosis, a mother cell produces two daughter cells with the same number of chromosomes. 2n from the mother cell produces two daughter cells with 2n. What about in meiosis? In meiosis, the mother cell is 2n, but it's going to produce four daughter cells that have half number of chromosomes. That is why you can see n each. So meiosis produces four daughter cells with half the number of chromosomes, while mitosis produces two with the same number. So mitosis, remember IP maths, I for interphase, P for prophase, M for metaphase, A for anaphase, and T for the telophase. Then of course there is cytokinesis. Please keep this in mind that the major part of mitosis is P-math, the prophase to the telophase stage. The interphase and cytokinesis are majorly not a part of mitosis, okay? So interphase is not part of the division process, but interphase is very important. It is important because it is in the interphase that we can refer to the interphase as the resting stage, yes, and the replication stage or the duplication stage. Why it is so called is because the DNA is replicated into two molecules or duplicated into two molecules. That is why interphase is very, very important. All right, now let's go to mitosis proper. In mitosis, we have the prophase, and for prophase, when we say prophase, we mean that some certain things happen, and to know the prophase, the chromatin is going to condense into the chromosomes. The cell membrane, as you can see here, will disintegrate. That means it's going to disappear, and then the centromeres, those green structures, are going to move to opposite poles to form the spindle fibers. And so these are the three key points that happens in the prophase stage. You should keep these things in mind. In the metaphase, the chromosomes are going to arrange themselves in the center or at the equator of the cell. And you can see that there's this line called the spindles. Spindles are attached to the centromere of each sister chromatid. The blue color and the red color are called sister chromatids. They are attached together. So this spindle is like a line. It is attached to the centromere of each sister chromatid. The spindles can attach to centromere because of the kinetochore protein. The kinetochore protein is important for attachment. Keep that in mind. And so in anaphase, the sister chromatids are separated. When they are separated, they become chromosomes, daughter chromosomes. So when sister chromatids separate, they become daughter chromosomes. These daughter chromosomes move towards the poles, the northern and the southern poles. And then the spindle fibers will begin to shorten. In the telophase is almost like everything that happens in the prophase stage, the opposite is going to happen. The daughter chromosomes finally get to the poles, and then the chromosomes will decoy or decondense to form tiny threads called the chromatin, and then the nuclear membrane will reappear. Almost everything that happened in the prophase in a reverse form. And so for cytokinesis, that means cell or cytoplasm division. In animal cell, the cleavage furrow is produced, but in the plant cell, a cell plate is produced. Please keep that also in mind. In meiosis is important for the production of gametes. It also causes variations, and it is divided into meiosis 1 and meiosis 2. Please do remember that in meiosis, four daughter cells with half chromosome number are produced, unlike in mitosis, where just two diploid cells are produced. But in meiosis, one diploid cell can give four haploid cells. Okay? And so in mitosis, for example, if we have 46 number of chromosomes, it's going to give two daughter cells with the same number of chromosomes, 46. But in meiosis, it's going to give four daughter cells with half the number of chromosomes, as you can see in this illustration. I hope you understand. And so for meiosis, formula is IP mass times 2. I'm going to explain why this formula has changed from the mitosis formula. In the interphase, after the interphase of the meiosis 1, we enter prophase 1, metaphase 1, anaphase 1, and telophase 1. Then we enter cytokinesis. Immediately after meiosis 1, we enter into meiosis 2, which includes prophase 2, metaphase 2, then anaphase 2, telophase 2, and another cytokinesis. That is why it's called IP math times. Like P math will be twice. That is meiosis 1 and meiosis 2. Okay, so let's look at prophase 1. Prophase 1 is similar to prophase in mitosis, but there are some exceptions you need to take note of. And number one is synapses, and the second exception is crossing over. Okay, so the next image is going to explain these two phenomena. What is synapses? What is crossing over in prophase 1 of mitosis? When we say synapses basically mean that two homologous chromosomes are going to lie side by side. Homologous chromosomes are a pair of sister chromatids that look alike. They are not necessarily the same. We can see two sister chromatids lying side by side together to form the homologous chromosomes. The purple color sister chromatid and the pink color sister chromatids. That's why they are called homologous chromosomes. But when we look at the different colors like pink and purple, they call them non-sister chromatids. So when they lie side by side and they cross over at the chiasma, that's the chiasma is the point where they will cross over their genetic information. We say crossing over has occurred. So synapses is lying side by side, but crossing over is exchange of genetic information. After they have finished crossing over their genetic information, they form the recombinant chromatids, as you can see here. We can also call them the variated homologous chromosomes because they have undergone variation. And after prophase 1, we enter metaphase 1. The recombinant sister chromatids will align randomly at the middle of the cell. Take notes, metaphase 1 is also a cause of variation. So crossing over and random alignment of sister chromatids causes variation. In metaphase, you can see that the variated chromosomes or the recombinant chromatids will align perfectly at the equator or at the center. In anaphase 1, the homologous chromosomes will separate and then are going to move towards the poles. In telophase 1, they are going to reach the poles and decoy to form the chromatin, and then cytokinesis is going to happen again, but this time two haploid daughter cells will be formed, and they're going to enter into meiosis 2 without going through interphase. In meiosis 2 is basically like mitosis. So the two haploid cells will become duplicated into four haploid cells after the normal prophase 2, metaphase 2, like that until you get to telophase 2 and cytokinesis. Then they will produce four haploid daughter cells, and that would be the end of meiosis. So at the end of meiosis, four daughter cells will be produced and they can differentiate into either the sperm cell or the egg cell. The genetic code. Genetic code is accumulation of 64 universal code words that instruct the cells to either stop or make amino acids. Okay? So out of those 64, 61 codons will make amino acids, but the other three are going to stop the production of amino acids. And so we call them stop codons. They are UAA, not AAU, please. UAA, UAG, UGA. All right. So this is the way the genetic code looks like. You want to look at the first letter, the second letter, and then you look at the third letter. There are three basic letters you have to focus on. So I'm going to label the third letter in 4 lines. Line 1, line 2, line 3, and line 4. For the first letter, we'll pick U. For the second letter, we'll pick U. And for the third letter, we'll also pick U in line 1. And we're going to form U, U, U. UUU codes for phenylene. So again, U, another U, and then the second one is C. We're going to form UUC. UUC also codes for phenylene, okay? Again, U, but the third line is A. So we have UUA. UUA codes for leucine. And so that is basically the way you are going to be interpreting the genetic code. But even if you do not understand it, don't worry. You can see that CAU codes for histidine amino acid. Just be looking at all of them, you will see that the amino acids are all labeled around them. Like AAU, for example, that is asparagine. We are going to use the genetic code to translate some information in the messenger RNA into amino acids. AGC from the genetic code is serine, UGU is cysteine, ACU is threonine, ACA is also threonine. When you look at the genetic code, you see that we have 4 amino acids from this sequence, okay? So it's not hard. You can check your genetic code. Let's try and transcribe these, or translate rather, this long information. AAU is, we already know the answer. What is AAU? Yes, asparagine. Next codon is UAA, and UAA is we'll check. I will not tell you. What do you think it is? It's nothing. Yes, because it is a stop codon. You will remember we talked about 3 stop codons, and because of that, the translation is going to stop. We're just going to have only 1 amino acid in this long sequence. Any of the 3 stop codons could be anywhere in the sequence, and immediately you get to a stop codon, you should stop translating. That's the key. Epigenetics means above genetics. So when we say epigenetics, every factor that manipulates gene expressions outside the normal DNA sequence. For example, let's say a set of twin boys, they have the same DNA, but there's genetic disease in one, and the other one does not have any genetic disease. How are we going to explain this? The only way is for scientists to study things like methylation outside the field of genetics. So we call it epigenetics.